Hi, I'm Brett. For those of you who aren't in my class, I'm the lab tech at PCC Cascade, and today I'm going to be setting up some Brassica rapa, also known as Wisconsin fast plants. And these are quick-growing plants that we use to demonstrate genetics. If you were doing this in the lab, you would be able to grow some plants, cross them, collect the seeds, and determine the phenotype of those seeds, all in the space of a single term. And you would be doing all of this yourself, but because we are remote this term, I'm going to set it up as a demo, and I'm going to, I've actually got some seeds so I can show you the parental phenotypes that were crossed, and then the F1 seeds that would have been produced from those parents, and then the F2 seeds that would have been produced by crossing, self-crossing the F1 seeds. I can plant those all at the same time and show you all the phenotypes that we get at the same time. So that's what I'm going to be doing. Some of the supplies that we're using, this is the watering system. It's just a plastic storage tray that we've cut a hole in the lid so that we can have this wick pick up water from the the watering system. That's the word I'm going for. On top of the watering system, we are going to place these quads. We'll break them up and have different plants, different seeds in these quads. Actually, maybe I'll do that for you now, just to show you how that works. These are little styrofoam pots, basically, that hold a little bit of soil, and then we can put in these pots a wick, which will wick water up from the watering system into the soil, so that our plants don't die. The other things that we're going to need for this, we need some labeling tape and Sharpies. We need the different seeds that we're going to plant. We need some soil. I'm just using kind of a seedling mix for potting soil, and some fertilizer. So that's what we've got. And then we're also going to need a light bank, and we don't have our fancy adjustable light banks available this term, but I did bring home a little LED light from the labs, and with a little creativity, I'll show you what that looks like. So there's our LED light, and it's in a cardboard box. There's another cardboard box inside so that we can adjust the height of those watering plants, or the watering system, in relation to the lights. So as the plants grow, we'll drop that further and further down. And then there's just some insulation material to keep them nice and warm as they're growing. Okay. So the first thing I need to do before I can plant anything, I need to label the quads. So I'm going to do that. I'm going to pause the video because that's going to be boring, but I'll pick up after these are labeled. Okay, so now that we have all of our quads labeled with the different seeds that we're going to be planting in there, it is time to, I'm going to make this a little brighter. It is time to get them set up with the quad wicks and the soil. And we'll also get our watering system set up with the water. And then once everything is all ready, then we'll plant the seeds. So we'll start with the quad wicks. To put those in the quads, just drop them in. Make sure they're sticking out from the bottom. And you'll need 4 of those in each quad. And again, I won't do all of them right now, but you get the idea. Then we're going to add some soil, but this soil fresh out of the bag is pretty dry, so I'm going to actually get some water in here before we try and add it to the quads. Actually, I'm going to let this water sit for a minute or two so it can kind of soak up into the soil. While that's doing that, I'm going to get some water into the watering system. And before I add the water, I'm going to add this anti-algal square, which will discourage the growth of algae, especially on the wick, but it's not going to prevent it completely. So, and this just has a little bit of I think, copper sulfate to try and keep the algae at bay. And then the other thing I'm going to do is I'm going to soak the wick in that water to get it good and wet before we put the quads onto it. Okay. And one tip I like to use, if you use warm water, that's going to help the seeds germinate a little bit better. Okay, so while that's kind of soaking there, let's see, yeah, I think our soil's pretty good. Actually, I'm going to bring the camera a little bit closer and zoom in to how we set the quads up. So let's see if I can zoom in. Okay. Yeah, like right about there. Okay. I'm gonna put this on this tray so we don't make a big old mess. Okay. So there's the quads. And actually, I'm gonna dip the quads in that water just for a minute. And that'll, the wicks will kind of pick up the water in there, so the wicks will start out wet. Like that. Okay, now we're going to add some soil to these quads. And the tool I like to use for that is just a chemical scoop. We've got the soil pretty well wet. Don't fill the quads all the way up when you're doing this. If you do, then they will be there'll be no room for the fertilizer that needs to go in. Maybe somewhere around half full for the quads right now. Okay, so that, if you can see that, that is the soil ready for the fertilizer. And fertilizer we use is just kind of a general purpose slow release for fertilizer called Osmocote. I'm gonna put some of that in here. and then probably about 3 pellets of osmocote into each quad cell. Okay, and this will just give the Brassica a little bit of nutrients as they're growing, because there's not a lot of soil in these quads. All right, now that we have the soil plus fertilizer, now we're going to add more soil almost all the way to the top of the quad cells. And again, if this soil is wet to begin with, it'll really help those seeds get started. Okay, so there is a quad that is ready for seeds. So I'm not going to show you doing this to all of the quads, but I will do this for all the quads, and then I'll show you how we plant the seeds. All right, now we have all of our quads with soil and fertilizer. On the bottom are the wicks, and when you put these on the watering system, you want to make sure the wicks are in good contact. The quad wicks are in good contact with the big felt wick. The other thing is you want to put them towards the front of the watering system, where the felt wick is picking up the water. If you put them towards the back, they get less and less water, and in the back they can dry out pretty easily. Okay, so now it's time to plant some seeds. It's critical that you make sure you're planting the correct seeds in the correct quads. The quads in front here are going to be the parental combinations, so I'm going to have some wild type seeds for exercise 8.1. The wild type have a purple, which is produced, it's a pigment called anthocyanin, so they have all the genes to produce anthocyanin. And then the other parent that we're going to be crossing, or kind of demoing the cross, is a non-purple parent, so it's not going to have the right genes to produce anthocyanin. And there's a particular allele called the anthocyaninless allele, ANL for short. So these, the wild type plants are going to have the dominant version of that allele, and they're going to be homozygous dominant, and the non-purple parents are going to be homozygous recessive. Okay, so I'm going to plant the wild type, the dominant allele, in 2 of the 4 cells of this quad, and I'm actually going to do the same for 8.2, and I'm going to plant 4 seeds per quad. Normally, if you were doing this in the lab, you'd only plant 3, but I think it's probably better to have an extra one just in case any of the seeds don't germinate. The seeds are tiny, so you want to handle these very carefully. And you can maybe see those in my hand there. And again, sometimes the chemical scoop is helpful for kind of counting them out and then planting them. So I think I've got 4 on that chemical scoop. I'll put them, double check they're going into the wild type quad. They are. And I'm going to do another 4 into the other wild type quad. And while I'm doing this, I'm going to do 4 into the wild type quad for 8.2. And then the other wild type, I should say quad cell, not the whole quad for these ones. Okay, so those are all our wild type parent seeds. And then I will do the the parents for 8.1, which is again that anthocyanin-less trait, and they are homozygous recessive for that allele. Let's see, and use my scoop to get just 4 seeds into each quad, or each quad cell. I keep saying quad instead of quad cell. And the extra seeds I will put back in the envelope. All right, so there's the parents for 8.1 are now all set up. And then for 8.2, this is a dihybrid cross. So we're looking at that anthocyanin-less allele. And then at the same time, we're going to be looking at another mutation, which is a yellow-green leaf. So these parents are homozygous recessive for both of those traits. So they're going to have a non-purple stem and a yellow-green leaf. And I'm going to plant those in as one of the parents for the 8.2. And again, this is just going to be 2 of the 4 cells. All right, so we'll get 4 of those seeds. Make sure they don't drop into the wrong cell. And there we go. All right, the extras back. Okay, so now our parents' quads have been seeded. Now we're going to put seeds in the F1 quads. So for the F1 seeds, if these parents were to grow, we crossed them and made sure that they didn't get self-crossed. And we collected those seeds. Those would be the F1 seeds that we're going to plant. And these are the same F1 seeds that we would give you in the lab to plant. Usually in the regular lab setting, we'd plant the parents ahead of time, then give you some F1 seeds to plant, and then you would have to grow those, self-cross them, and collect the seeds to plant for the F2. So these we buy from Carolina Biological. And for the 8.1, again, that's the monohybrid cross, just looking at that anthocyanin-less trait, or I should say the anthocyanin gene, really. And this package maybe gives you a hint about what phenotypes we should see in the offspring, but we'll record the phenotypes later on after these plants have grown up. So I'm going to get some of these seeds out. And again, I'm going to do 4 per cell. And for this quad, I'm going to do every cell with the same seed. Come on. There we go. All right. So the seeds for 8.1 of the F1 generation have been planted. And then the 8.2, we'll do those. And again, these are F1 seeds, so they're going to be the cross that would result from these parents. But these are going to have the non-purple, or they're going to be dihybrids, right? So for the non-purple stem and the yellow-green leaf trait. So these are going to be theoretically heterozygous for both of those traits. Don't have many of these seeds left, but I think we've I think we've got enough. All right. So those are in. All right, so the last set of quads to plant is going to be the F2 generation. So again, for exercise 8.1, these are the seeds that have been collected from the cross or the self-cross of that F1 generation. And for these, we have some extra seeds that were collected by the Biology 212 students during winter term. And so hopefully we can trust that they did their crosses correctly and collected the right seeds into the right envelopes. We will trust them on that. And again, we're going to do 4 seeds per quad for the 8.1 exercise, and then we'll do another 4 seeds per quad for the 8.2. So we've got some of those seeds as well that were collected last term. Alright, put the extras back in the envelope, and we'll plant the 8.2 F2 seeds. All right, so we have 4 of those seeds in each quad. All right, so now we have all of our seeds in the soil. There's one last step before we put this under the lights, and that is to add a little dusting of dry soil right on top of the seeds that we just planted. Just gives them a little protection, helps absorb some heat from the lights, and so they tend to grow a little bit better that way. We'll get the bag of soil back. Just a light little dusting on each cell of each quad. This soil's maybe a little damp to do this, but yeah, we'll make it work. Don't want to add too much soil here, because then the seeds might not have enough energy stored to get all the way through that layer. It does work a little bit better than just the bare seeds on top of the soil. soil. Okay. There's a little bit of extra soil now on all of them. And these are almost ready to go under the light banks. I just need to get one more thing. I'll be back in a minute. Okay, the last thing that I want to do to these Brassica seedlings, or they aren't seedlings yet, Brassica seeds, is give them a little extra water just to kind of get them started. And really for the first couple of days that they're growing, we want to be sure to water them regularly, and I'll probably water them at least twice a day for the next couple of days. So it doesn't have to be deionized water. Tap water works just fine, but I happen to have this in a squeeze bottle, so that's kind of the key. And you just go and water each cell gently. You don't want to add so much water that the seeds end up coming out of each cell. But you do kind of want to make sure that soil gets nice and saturated. All right, so now these are ready to go into the light bank, and I will set that up with the light, and I'm actually going to set that up in a spare closet here at my quote-unquote lab. So yeah, put them in there, and again water them a couple times a day for the next 2 or 3 days. And once they start germinating, I will show you what that looks like.